the next walkthrough is going to be the enzyme lab, and that's going to be the first one that's going to be conceptually a little bit interesting. So the walkthrough is going to be right here. And again, you've got a lab handout. In the lab handout, this time, again, you just click on it and you get to the lab handout. In the lab handout this time, we actually have pictures. We talk about the scientific method again. We talk about how enzymes work and how these tools work. And in biology, everything has to do with shape. And so what we're doing is we actually isolate an enzyme. And the enzyme is actually responsible for the browning of potatoes. And I go over that in the lecture. And what we would be doing in the lab is we would literally be chopping up a potato, blending it, and isolating the enzyme that way. And so the enzyme is right in there. And then what we're going to do is we're going to look at the activity of the enzyme and how different environmental things change the activity of the enzyme. And by doing so, we learn something about the nature of the enzyme. So specifically, we're looking at the enzyme specificity. So by looking at three different chemicals that are shaped very similar to the substrate catechol, we look at whether the enzyme reacts or doesn't react. So it turns it brown or doesn't turn it brown. And as you can see, there's very different reactions in that. We read this with a spectrophotometer, and so we talk a little bit about how a spectrophotometer works, and how what the numbers mean that we see there. And then you look at other different, so in the lab, we would then incubate this enzyme at different temperatures. We look at the effect that temperature has on the enzyme activity. So you need to think about what this means, what these temperatures mean, right? What's 40 degrees Celsius, what's 60, what's 100 degrees Celsius. You got to have a little bit of an idea what that means in relation to Fahrenheit. Then there are some questions, longhand questions that are there. Again, go look at the quiz if you have no clue how to even answer this question. Go look at the quiz, and it gives you sample answers that you may be able to pick. And then then we check on the pH, if pH has any activity on enzyme activity, and then you're doing some graphing yourself, but I'm not going to have you turn in this graph. There's two of them, and then last but not least, where is it? Oh, where is it? Hang on. Oh, that was in the other one. Never mind. It wasn't the enzyme lab. Never mind me. The labster in this case. So we talk about the shape of proteins and how they function in the background. Here I talk specifically about how the lab works, how the spectrophotometer works, and then as usually you have a quiz. This labster activity is a bit of a bear. Don't worry too much about Michaelis-Menten and any of those kinds of things. We're not going to worry about it. I talked to you in very basic terms how to think about enzymes and on how to think about enzyme activity and how to measure it. Look at my videos because I'm trying to explain it to you in terms of a tool as a very, and I think of enzymes as a tool in terms of a turnstile that allows people to go through, and it works pretty well on a very basic level to understand what happens if you add more enzyme, what happens if you add more substrate, and those kinds of things. So I want you to think a little bit about that. And then you have the handout question set, which then allows you to put in the answers to the handout question set. And that's it for this week for the enzyme lab.